Hello, everybody, and welcome to the Genetic Medicine Evolution Revolution. Today, we will be walking you through the discovery and early preclinical session. My name is Kira, and I am the product manager for Ignite. I also have my colleague Angela Zhang on the line here as well, and she will be walking you through some of our reagents and our Spark offering. Next slide, please. Great. So there are many pillars to a successful genetic medicine, and designing and developing each of these pillars requires a fully different skill set. So this is not an easy challenge generally. The payload, your delivery of your nanoparticle, as well as your manufacturing, these are all unique challenges that you face when developing a genetic medicine. Next slide, please. On top of that, there's also different challenges that you face when at different stages of development. So in drug discovery, in clinical development, and in your approved drug process, you have a bunch of different concerns that you are trying to navigate and satisfy. All right, next slide, please. So this is how we've kind of with these challenges in mind, this is how we've come up with our NanoAssemblr suite of products to really enable this genetic medicine development. So we have a range of instruments that take you from early drug discovery and screening through to early preclinical development into late preclinical development, all the way through to commercial manufacture. And the way that you work through this pipeline, sorry, previous slide, please. And the way that you work through this pipeline is generally you start with a large number of potential drug candidates when you're doing your screening activities, and you slowly narrow those down as you go up in volume and progress through this pipeline. So that's really what we are going to talk about today, is how you start at the very earliest stage here in discovery and screening, and then how you move through into your early preclinical development. And one of the ways that we have really enabled this is that we have developed our next-gen technology. So this is a very precise microfluidic mixing method, and it's completely scalable from, again, very, very small scale all the way through to GMP commercial manufacturing. All through a single mixer, which is pretty exciting. You can make volumes from 250 microliters all the way up to 50 liters. So that's very, very cool technology that is fully innovative and developed by P&I. Next slide, please. Another tool that we're leveraging here is a deep understanding of the unit operations that are needed to create your final drug product. So whether it is from your microfluidic mixing into your inline dilution or your TFF, all of these unit processes are considered and scaled appropriately for the stage being worked on. For instance, on the Ignite system, we do have inline dilution, which we carry through all of our systems. So these are tools that you can really start to study and develop a deeper understanding of at the earliest stages. And with that, I'm going to hand it over to my colleague, Angela, to tell you a little bit more about the Spark.
Great. Thank you so much, Kira, for that great introduction. So as she already alluded to, it is very important, when it comes to genetic medicine, especially during these early discovery, a preclinical stage where you're able to screen a large number of candidates in a very small volume, exactly as what Spark would allow you to do. And the formulated LNPs at this volume can be introduced into in vitro cell cultures or in vivo in a small number of rodents. And here you'll start to really see the intrinsic benefits of the LNP technology, where it utilizes an intrinsic cellular pathway for entry. LNPs mimic low-density lipoproteins, or LDLs, which bind to the LDL receptor on the cellular membrane and is internalized via endocytosis. They are pH sensitive, so they are designed to release their payload into the cytoplasm. Now, we designed Spark to really ensure that you can be creative with your genetic medicine, your LNPs, It is a robust delivery system where you get efficient encapsulation and consistent particle size reproducibly. Because you're also working at the microliter scale with your starting material, you can really save on your expensive or limited starting materials, especially at this early stage where you likely will experiment or try out many different conditions. So that's a really great thing. You can also save on time, as I will show you in the next slides or so. The formulation can really be completed within seconds. So that's really how quick and easy it is. And finally, when it comes to, and this is very important when it comes to comparing with other methods of delivering genetic materials into cell cultures, is that you're in the case of Spark and using the LNP system, you're really applying the formulated LNPs directly onto your cells in the cell culture. So you're really not doing a lot of extensive, centrifugation, resuspension, not putting stress onto your cells. And again, you get the higher cell viability and again, saves your time because you're not doing a lot of manipulation of cells. So that's really important. Now, when you look at the workflow with Spark, it is incredibly simple. You're pipetting your nucleic acid, you're pipetting your lipid mix, for example, using our pre-made reagent product and a formulation buffer into the cartridge. You put a cap on it, you insert it into the machine, you push start, and in less than 10 seconds, you got your formulated LNPs. That's it. You pipette the particles out, you dilute them, and they're ready for downstream assays. It is really that simple. Now, because we make this so straightforward at this stage, you can really focus on the design of your products. When it comes to the payload, what would that look like? When it comes to the carriers, I mean, if you're interested in exploring on your own, there's a lot of different considerations. Now, we also have, again, reagent products or formulation solutions. If you would like us to work together on that, we can certainly do that as well. But really, downstream from there are hundreds of possible formulations and experiments you can do with, a very small amount of starting material. And that's the strength of Spark. Okay, so now I'll pass it to Kira to talk about Ignite.
Great. Thanks, Angela. Next slide, please. Great, so we just finished talking about Spark there. So that is the lowest volume offering that we have. And the next stage in this is really the next stage up, it's the Ignite. So this is 1 milliliter up to 20 milliliters. And this is again where you can start to develop your formulation in more detail, as well as optimize it for the best delivery. Next slide, please. So to do this, we've designed Ignite. And Ignite is a very, very easy to use system. So it doesn't take much time to learn how to use this system, and it also doesn't take much time to actually perform a run. So very easy to use. It's also very reproducible. So it doesn't matter your level of expertise in running this. It doesn't matter what day it is. It doesn't matter anything like that. It's very, very reproducible. There's very little variability between your runs. It's also entirely scalable, which is another great benefit of Ignite. Whatever you develop here, you can take further on down the pathway into Blaze and GMP manufacturing. So Ignite really is that bridging that gap between this discovery phase into the early preclinical phase. So taking what you've developed on Spark in your early screening experiments and developing your formulation further. So whether it's your excipient identification through into your formulation development and optimization, Ignite really is the tool for that. And it's because it is so reproducible, you can use this make, test, and select process, this very iterative approach to your formulation design and development to really, help you speed up that process and perform a very controlled set of experiments. Next slide, please. So as I said, some of the key differences between Spark and Ignite, we can talk about volume. Obviously, volume is a big difference here, up to 250 microliters versus 20 milliliters. But even beyond that, there are quite a few differences in terms of what these instruments are designed to do. So the Spark is designed to be very, very simple. There's one button, there's very little control. It uses air to actually push your fluids through the microfluidic mixer, the next gen mixer. So there is no control over flow rate ratios or total flow rates or waste volume. It's all the exact same every single time. So you're not necessarily trying to optimize your formulation here. What you are trying to do, though, is screen through large numbers of samples to understand really your first proof of concept, what's working well and what's not. Then you can take that onto the Ignite system. And this is, as I said, again, where the real development and optimization occurs. So Ignite now, it uses syringe pushers to very precisely move the fluids through the microfluidic mixer. And that enables you to very precisely control your total flow rate and your flow rate ratio, as well as your waste volume. So these are all critical instrument parameters that we use to optimize the formulation. Next slide, please. And because of that, as I said, it is very controlled and reproducible on the Ignite scale. So some of the instrument parameters I just mentioned, total flow rate ratio, and your waste volume. But beyond that, you can also dive into your chemical or your chemistry parameters as well. So this is where you can start playing into even in more detail with your formulation and how it performs. And again, just because of the nature of the microfluidic mixing with our next gen mixer, it is entirely reproducible. So you can take a very systematic and iterative approach to your formulation development and design. So you can start to control for your variables as you're moving through this process and slowly narrow down your selection to your lead candidates. Next slide, please. So Ignite has been designed with the end in mind. And this, as you can see from some of those opening slides, is very critical to always be thinking about what that end goal is. So what is your final drug product and how do you get there? Because your considerations are changing throughout your early development all the way into your clinical development, into your commercial manufacture, you really need to sort of simplify that whole process down into your key unit process steps. And for genetic medicine, these are the steps that we've identified as the key process steps to get to that final drug product. So these are the things that you'll have to perform every single time, no matter what scale you're at, to get that final drug product. So we've tried to implement as many of these as possible on Ignite to facilitate, the early study of these process parameters and to understand their effect on your final drug product. Next slide, please. Ignite has also been designed for the future. So we do have a third inlet on Ignite. Generally right now is being used for dilution. Again, one of those key process steps that we want to investigate. But generally you can also consider what this may enable you to do in these new sort of nanoparticle types that have yet to be developed. It really does add this whole other parameter space that we can now start to play with and understand how that impacts our particles. Next slide, please. Which means that we already support a very wide application space for our NanoAssemblr technology. So from LNPs to liposomes to polymeric nanoparticles, all these different particle types, you can now imagine what this third inlet stream will start to enable us to support as well. The list really is potentially endless, so it's very exciting. Next slide, please. And also Ignite, as I said, is very easy to use and it's very fast to use. So not only have we sort of designed some very unique usability features such as recipes. So that is a set of formulation parameters that you know work and you would like to save for future use. You can now save those on the instrument. We also have histories, so you can see all the runs that you've previously done and you can, with one button, run them again. But beyond that, we also have a single-use cartridge. So this is a huge time saver. It probably cuts your total formulation time in half. Next slide, please, here, Angela. So not only is a single use a huge time saver, it actually is a much less risky environment to perform your formulation in. So the single-use cartridge really ensures that your fluids are being introduced in the most consistent manner possible. So the chance of any leaks or of any aggregation or anything like that is greatly reduced, and it really does ensure that your nanoparticles are very consistent every single time. As I said, it also is very fast. So that is, it takes about half the time to run this. You put your cartridge in, you run, and you're done. Everything is disposable. And it also does, sorry, previous slide here, Angela. It also does increase your flexibility. So if I have multiple users on the same platform running potentially very different formulations, I can very easily switch between these programs with no worry of contamination or cleanup or any prep involved. And actually pretty counterintuitively, the single-use model actually does use less plastic waste than the previous multi-use cartridges do. And that is generally because when you're using this multi-use cartridge, all of that cleaning and that priming that you need to do with new syringes every single time, those syringes are actually a very, very significant amount of plastic. So minimizing the amount of syringes that we're using is actually saving a lot of plastic waste in this situation. Next slide, please. And of course, both the chemistry and the process affect the outcome. So we've talked a little bit about Ignite here, but we're talking about these pillars of genetic medicine development, your payload and your delivery system are also critical considerations here. And that's really, when you consider all of those things, that's how you end up with the most homogeneous LNPs. So I'll hand it back to Angela here to talk a little bit about some of our delivery technology.
Great, thank you so much, Kira. That's really well said there. Let me just go back for a second about both the chemistry and the process affect the outcome. So I'm going to talk a little bit more about the sort of chemistry side of things, and specifically about GenVoy-ILM or GenVoy-R delivery system. And essentially, this is a pre-made lipid mix that enables you to prepare your own LNPs together with the NanoAssemblr platform. So what this really does is allow you to start with the right building blocks to take your genetic medicine from early discovery through preclinical studies. Now we can talk about how we can help you take your genetic medicine all the way through clinical and commercialization, and we're happy to provide more information on that. But what we're really trying to do here is that we want to make genetic medicine accessible with this delivery system, easy to use and scalable. And that scalable part is something Kira already started talking about with Ignite, and you're going to hear more about that from our colleagues later in the day. Now, GenVoy-ILM has been used in many different applications for discovery, to support discovery and preclinical development. For instance, gene therapy, cell therapy, and vaccines. It has also been used to manipulate genes in a variety of different ways, silencing, expressing, editing, and modulating. And it's been used to encapsulate a wide range of genetic materials. Now, I want to show you some proof of concept studies we have done with GenVoy and with our NanoAssemblr platforms in the next few slides. Now, one case study around the discovery of a new therapeutic target. Here what we did is to perform a proof of concept study to knock down HRPT gene using siRNA in primary rat cortical neurons. And what you can see on the left here is that you're seeing significant knockdown of HRPT gene in a dose-dependent manner. And on the right here, when you look at the viability using a fluorescence viability assay, what you can see is that there's really not much change in cell viability when you compare the treated conditions to untreated. So there's really not any observed toxicity to these cells. If you recall what I said earlier about the SPARC system, I think that's really the strength, and also the intrinsic nature of the LNP technology. This is really a huge advantage when it comes to cell viability. And of course, really nice knockdown here. Now, moving forward from discovery, we also performed a proof of concept study to really benchmark that therapeutic performance. And here we utilize an in vivo model anemia and deliver erythropoietin or EPO mRNA formulated in LNPs. So what you can see on the left here is, and in this study, we actually did it comparatively between, our GenVoy and the formulation formulated with a clinically approved lipid called MC3. So what you could see on the left here, we actually saw very comparable particle size and polydispersity between the EPO mRNA formulated in GenVoy and MC3 formulations. And on the right here, when you look at the functional readouts of these delivery, you could see increased erythropoiesis upon the delivery of the EPO mRNA-LNPs, and this is comparable between those formulated in MC3 and in our own GenVoy. So that's a very exciting result to see. Now, when we take this model further to utilize it as a proof of concept to develop process for scale-up, what you can see here is that on the left, where you can see really consistent size and PDI, again, amongst the EPO mRNA formulated with GenVoy from a small scale to increasingly larger scales, starting with Ignite, which Kira talked about, but also all the way through Blaze and GMP, which you will hear later from our colleagues today. And this is also true with encapsulation efficiency. Again, very consistent performance from smaller scale to larger scale. And when we look at the functional readout of that, and in this case, we looked at the total serum level of EPO, and again, the increased erythropoiesis on the right there, you can see once again, there's increased protein expression and function consistently, across all scales of development from Ignite to GMP, and really demonstrating the scalability of the formulation and the process. And again, you will hear more about the power of scale up from our colleagues later today, but I thought I could give you a sneak peek here. Now, after all that, I'm sure you're excited to see Ignite with GenVoy in action. So today we have Kira in the studio to give you a demo of Ignite with GenVoy. Now, before we get started, I just want to quickly show you some considerations when it comes to preparing mRNA with LNPs for an in vivo study. Some of the considerations include, what is your in vivo dose? Now, typically, there's a range for mouse. So you have to think about how many mice do you want to have to see that statistical and biological differences. You also need to think about how much material to generate, and that really depends on, how many replicates you're doing. Are you doing multiple doses? Are you looking at a dose response curve? And always prepare a little bit of extra for characterization and things like that. And finally, what is the quality of your payload, right? That depends on the design, and also purity is very important. So with that, I will pass you to Kira, and who will get you started on the demo.
Great. Thanks, Angela. All right, so as Angela mentioned, I am in our lab today, and I will be running a GenVoy formulation. But first, let's just take a quick overview of the instrument itself to understand what we're working with here. So this is the Ignite instrument, as you can see here. Spark is in the back here, so you can see the size difference here. Ignite is fully self-contained, though. So there's one plug at the back. There's an onboard touchscreen to control it. There's no other wires, nothing else to clutter up your bench space. So really, we tried to minimize the footprint of this device so that you have as much room to work around it as possible. At the front, in this lid here, if we open it up, you can see that we have 3 pushers. So these are what it will actually be interfacing with the syringes that we load with our reagents. And these are what will depress the syringes to flow through the cartridge very carefully. So each of these has a very precisely calibrated and tuned syringe detection switch on here. So that is how we ensure that the fluids are actually entering and meeting appropriately within the cartridge. And maintaining calibration on these is very important to ensure that your waste volumes are minimized. Up here, you can see we have what we call the rotating block. So this actually rotates up and down so you can see the cartridge lures quite easily. It is labeled with an L, C, and an R, so that that's how we refer to the channels. Dilution is always on the L channel, and C and R are your aqueous and your organic, respectively. So underneath the rotating block, we also have this, on this instrument at least. So this is our optional heating block accessory. So this can be purchased in addition to the basic Ignite instrument. And what it is it is 2 separate syringe heaters. So if I do want to perform a formulation at some temperature above room temperature, up to 75 degrees Celsius, this is what I would use back here. Both of the syringes can be heated independently. So that is, I can have one at a high temperature and one at room temperature. So it really does allow you to play around with that new parameter. So then over here, you can see that we have the sample and the waste collection. So this is actually just an arm that you can pull out and clean and replace if needed to hold your Falcon tubes to collect your sample and your waste. Again, it's very clearly labeled over here. Now, if we move over here, this is the screen that controls the whole instrument. So we have a few different features on here. Generally, we have what we call a quick run. So quick run is probably what you're most familiar with. This is what you would use if you just want to try out some parameters, do a quick run, see what happens, but you don't know whether these parameters will work yet. So here is where I would start to enter, the syringe size that I'm using. For instance, a flow rate. This is a flow rate ratio here. So let's say, let's just pretend this is a 2-to-1. Total volume, let's say I want to run 5 mils. Total flow rate here, we can say 12 mils per minute. So very quick, very easy to use and to enter parameters in here. Start waste, I can put some value in here, and end waste as well. So very, very easy to do on this touch screen, and very familiar technology. We also have, as I mentioned, recipes. So if there are a set of parameters that I know work and I would like to store them, I have 10 recipe slots that I can use to do that. So here, if I have a recipe saved, I can just go ahead and run it very, very quickly, or I can go through and edit it. And this looks very similar to the quick run screen. The only difference here is that I can now name it as well. So recipes are a very useful tool, again, to speed up our workflow as much as possible, as is this history. So in the history, I can view up to 800 runs of my previous runs. Not only that, I can also see whether they were a success or whether it was incomplete, whether it was stopped for some reason. I can also see whether it was a recipe that was run or a quick run, and it gives me some basic parameters as well as time and date. So again, very easy to use. In my settings over here, I can, again, change the time and date, or I can also change the syringes that I'm using. So we do allow use with 3 different syringe brands and 4 different syringe types. So we have the BD syringes, we have the B bronze syringes, and we also have Terumo syringes. So if I wanted to start using bronze syringes, I would just click these and they would then show up in my pop down in the previous page. So again, very, very simple, and that's all there really is to the interface here. So then let's run some GenVoy. So what I'm going to do first is open up a cartridge. So this is the cartridge here. As I said, they're all single use. They're all individually packaged. So I would just take the cartridge out of the case there. I would put in, this is the cartridge adapter. So this is what we use if we're using just a 2-inlet cartridge. The cartridge adapter just snaps into place over here. So now I'll just slide my cartridge in like this. Just clicks into place, a nice soft click, and we're ready to go. So I'll rotate this up so that I can now see my lures quite clearly. So it is always important to get a good syringe fit in your lure so that you minimize any leaks. And so today I will be running a 2 milliliter total volume formulation. So I'll start by filling a 3 ml syringe here with my aqueous phase. So I'm just putting a blunt tip needle on this here so that I can go into this tiny vial quite easily. So you can see that this vial is quite small, which this needle will help me get in there. Oops. There we go. So first things first, I'm just going to pull a small volume in here first. It is very important to make sure that you remove any bubbles in the syringe because these can impact, again, how your fluid streams meet, and that can ultimately impact the reproducibility of your particles. So here you can see I have a big bubble, so I'm just going to slowly push that out into this Kim wipe here. Great, great. And then I can continue to fill this up. And again, getting rid of any bubbles in that process, just tap them to the top and slowly push out. Okay, so once I have this, sorry, I'm just finding the camera. So once I have this ready to go, I can just come back over to the lure here and twist the syringe into place. So when you're locking that into the lure over here, you don't have to over tighten. You just go until you can feel some resistance, and you don't have to keep twisting until you can't anymore. The more times that you put a syringe on the cartridge and remove it, you are loosening that lure fit every single time. So this is, again, why it's very important that we have a single-use cartridge to make sure that there is no leaks around the lure. All right, so that was my mRNA. And here I am going to fill up a 1 ml syringe, same sort of thing, with my GenVoy ionizable lipid mix. So again, I'll put my blunt needle on here. And so this is a GenVoy here. So today I am running the, I'm running our training kit, but we also do sell GenVoy separately on its own in larger quantities. So same process here is start by just taking a little bit in before then getting rid of any bubbles that I have. Okay. Great. And so with this, oops, sorry. With this 1 ml syringe, you can see that this is a lure slip tip. Again, with all 1 ml BD syringes, we recommend the lure slip tip as opposed to the lure lock because the syringe itself is made of a different material. So the lure slip in the 1 ml variety is much more compatible with the solvents that we typically use. So with the lure slip, I'm just going to push and twist into place, just like that, and then rotate this block back down. So as I said, over here, we do have our aqueous component, which has our mRNA in it, and this is our ionizable lipid mix over here. Some other important things to do here are put in your sample in your waste container. You would be surprised how many times I forget to do this. So I'm just going to quickly label my vial there and push this into place. And so it's important when these are going in that you push them in and then push them all the way to the bottom. So you want the threads as sitting as far down as possible. And this is just because when this moves out to collect your sample underneath the cartridge, you want as little drip space in there as possible, but you also don't want any interference with the cartridge itself. So it is important to push these down as far as possible. Okay, so now I'm just going to close this lid. And if we go back over here to the recipe, I already have a GenVoy-ILM training kit recipe in here. So just with one button, just press this green button here. And I'm then taken to this page. So this is where I can confirm that everything I've just done looks correct. So first, does my recipe look correct? So I'm going to make 2 milliliters of total volume. I have a 3 ml syringe and a 1 ml syringe loaded properly. We're going to run at 12 mils per minute with a 3:1 flow rate ratio. My waste will be 0.35 to start and 0.05 to end. That all looks good. My syringes are correctly locked into place. I have enough reagent in each syringe. That is correct. So it is always important to put a little bit more reagent in than you actually need, just to be safe. And then the sample and waste containers are attached. So everything looks good to go, and I will just go ahead and press start here. We could switch views as well. There we go. So you can see that the tubes have just moved into place. The pushers move up to find the syringes, and then they start depressing here. And there we go. That's it. So we just made 2 milliliters of mRNA-LNPs in a matter of minutes there as well. So now that this is done, I can open this lid here, rotate this block up, remove these syringes. Just go straight into the trash. So does this cartridge. If you see any drips like this, just give those a quick wipe down whenever you see them. And that's all that's needed. And now I can take my sample for some further processing. So what I will do now is I'll actually size this so we can make sure that our LNPs turned out correctly. So first things first, I'll grab my little cuvette over here. So what I will do is I will add 400 microliters of my formulation buffer. All right, and then I will add 40 microliters of my sample. Okay, give that a few little mixes in there. Make sure that there are no bubbles. And then I will go ahead and put this into our sizer over here. So let's run this and take a look. So this will start running here. And generally with this training kit, we expect the performance to be, I believe it's 60 to 120 nanometers and a PDI of less than 0.25. So that's what we're looking for here. So this will just take a few minutes. And there you go. So the first results coming in, it does look like our size is about 94-ish, 95. And our PDI is 0.08. So this will likely change over the next few runs, but that's a good indication that things are working. So what I've just done there is I've used the GenVoy-ILM, or ionizable lipid mix, as an off-the-shelf solution to encapsulate mRNA. So GenVoy is a great tool to very quickly and easily get up and running with LNPs and to start performing some of your early preclinical studies. And this is just going to continue to go, but yep, I did run an earlier run this morning and we had similar results. So it's looking like we're, yeah, around 90-ish nanometers and a PDI of roughly 0.1. Yeah, so with that, I'll leave it there. But if you have any other, questions or comments or would like to know more, Angela and I will both be in the booths following this presentation. So we'll be there to answer any questions that you may have had about this presentation or about other things in general. And we'd love to chat and get to know you. So thanks for tuning in, everybody.